All right, let's just get right into it. Have you ever actually stopped to think about the incredible microscopic world that makes you, well, you? Welcome to this explainer on the dual engine of life, the cell cycle and cell division. We're going to break down the biological machinery that literally powers every single living thing on Earth.
I mean, how does a single microscopic cell become you? It is completely mind-boggling when you really think about it. Every multicellular organism out there, including you and me, starts life as just a single lonely cell called a zygote. And that raises the ultimate biological question. How does that one tiny speck of life build an entire complex body made of millions of cells? And how does it fix itself when things inevitably go wrong?
So here's how we're going to break this down today. We'll start with the magic of one cell. Step into Interphase, what I like to call the prep kitchen, check out Mitosis, the cloning machine, jump over to Meiosis, the remix engine, and wrap it all up with the ultimate cellular showdown.
All right, part one, the magic of one cell.
Let's zoom out for a second and look at the daily routine of a cell, a sequence of events we call the cell cycle. A typical human cell takes about 24 hours to finish one full cycle. But here's the wild part. 95% of that time is just interphase. That's the cell simply preparing to divide. The actual physical division, the M phase, is just this tiny little 5% sliver of time. It's literally like spending all day cooking a massive Thanksgiving feast just to scarf it down in 5 minutes.
Moving right along to part two, Interphase, the prep kitchen.
So during that massive 95% window, the cell might look totally chill from the outside, but inside, it is hustling. We've got three main steps here. Step one is the G1 phase, gap 1, where the cell is just growing rapidly, synthesizing RNA and proteins. Then we hit step 2, the S phase, and this is crucial because it's where DNA replication happens. Finally, step 3, the G2 phase. This is where the cell puts the finishing touches on everything, stockpiling ATP and building the specific proteins it needs to actually pull off the division. Now, if you're taking notes for an exam, pay attention right here. During that S phase, the physical amount of DNA literally doubles. It shoots from 2C up to 4C. But, and this is a massive but, the actual chromosome number does not increase. It stays exactly at 2N. Why? Because it's not making brand new individual chromosomes. The existing ones are just duplicating their material to form sister chromatids. Think of it like photocopying a page in a book. You've got twice as much paper, sure, but the page number? Exactly the same.
You know, not every cell actually stays on this dividing treadmill forever. Some of them permanently hop off into what we call the G0 phase, or the quiescent stage. Think about your heart muscle cells or your neurons. They are still highly active, doing all the heavy lifting to keep you alive day to day, but they've officially hung up their dividing hats. They just tap out and maintain their current state. No more replicating.
All right, part 3, mitosis, the cloning machine.
For the cells that do keep going, their prep time ends, and they fire up our first biological engine, mitosis. We call this an equational division. It mostly happens in your somatic cells, your regular everyday body cells. Basically, one parent cell with a 2N chromosome number splits to create two identical daughter cells, both sitting perfectly at 2N. It is a flawless cloning process. Absolutely no genomic shuffling, no surprises, just exact copies doing the daily work of helping you grow and repair tissue.
So the actual division of the nucleus, the fancy term is karyokinesis, goes down in 4 highly coordinated phases. First up is prophase, where all that messy chromatin tightly condenses into visible chromosomes and the nuclear membrane just vanishes. Next is metaphase, where those chromosomes perfectly line up right down the middle, like a biological equator. Then anaphase hits, and this is the action movie part. The sister chromatids are violently ripped apart and dragged to opposite sides. Finally, telophase. The chromosomes decondense, and 2 brand new nuclear membranes form around them.
Once the nuclei are split, the rest of the cell body has to follow suit to finish the job. This is cytokinesis, and it's actually super fascinating how differently life forms handle this part. Animal cells? They have soft membranes, so they just pinch inward, creating this cleavage furrow until the cell eventually just snaps into 2. But plant cells have those incredibly rigid cell walls. They literally can't pinch, so they build a new wall from the inside out, a cell plate, growing it outward until it totally separates the 2 new cells. Pretty clever, right?
Okay, but what happens if a cell finishes dividing its nucleus over and over, but then just forgets to divide the actual cytoplasm? Well, you get a syncytium. It's essentially a giant multinucleated megacell. The nucleus duplicates, but the outer boundary never actually splits. Sounds totally sci-fi, but I guarantee you've encountered one. You know the tasty liquid inside a coconut? That liquid endosperm is a perfect, real-world example of a syncytium.
Before we move on, let's quickly recap the superpowers of this cloning machine. Mitosis is truly the unsung hero of your biology. It maintains a perfectly stable chromosome number across billions of cells in your body. It drives your physical growth. It's what patches up a scraped knee, replaces the millions of skin and blood cells you shed every single day, and even lets plants continuously sprout new leaves and branches. It is the ultimate bodily maintenance engine.
Which brings us to part 4, Meiosis, the remix engine.
Now, maintenance is great and all, but to create totally new life, we needed a completely different approach. We have to swap out the cloning machine for our second engine, Meiosis. This is famously a reductional division, and it's strictly reserved for your germ cells. Look at what's happening here. One 2N parent cell doesn't divide just once, it divides twice. It cranks out 4 genetically distinct gametes, sperm or eggs, that carry exactly half the original chromosome number, represented as just N. We're having the genetic material so that when sperm meets egg, boom, the resulting zygote is right back to that perfect 2N number.
Meiosis I kicks off with prophase 1. And let me tell you, this stage is a total marathon. It's incredibly complex, broken down into 5 intricate substages. It starts with leptotene, where chromosomes become visible. Then, in zygotene, something amazing happens. Homologous chromosomes, the matching ones you got from your mom and dad, actually find each other and pair up in a tight embrace called synapsis.
Then we hit pachytene. And this right here, this is where the absolute magic happens. While they're locked in that embrace, these non-sister chromatids physically swap pieces of their DNA. We call it crossing over. As they pull apart just a tiny bit in the next stage, diplotene, you can literally see the X-shaped spots where they traded genes, called chiasmata. This genetic swap is the biological twist that guarantees you are completely unique from your siblings. It is literally the origin story of human individuality.
After they line up at the center, the cell pushes into anaphase 1, and the twist here is wild. Remember how in mitosis, the sister chromatids were violently ripped apart at the centromere? Yeah, that doesn't happen here. In anaphase 1, the homologous pairs are separated, but the centromeres stay completely intact. The entire paired sister chromatid gets pulled as one single unit to the opposite pole. This exact moment is when the overall chromosome number officially gets cut in half. After a super quick breather called Interkinesis, where, by the way, absolutely no new DNA is replicated, the cells dive straight into meiosis 2. This is the equational follow-up, and it behaves almost exactly like our old cloning friend mitosis. Those sister chromatids that held on so tightly earlier, they finally get split apart at their centromeres in anaphase 2. The grand finale leaves us with 4 totally unique haploid cells, fully primed and ready for reproduction. So what are the superpowers of this remix engine? Why is meiosis such a big deal? Well, first off, it ensures chromosome stability across generations. By cutting the count to N, it guarantees your kids won't end up with double the chromosomes you have. It obviously produces the gametes for reproduction, but really, the most profound thing it does is generate immense genetic variation through crossing over. That variation is literally the raw material that fuels natural selection and drives evolution over millions of years.
And finally, part 5, the ultimate cellular showdown.
All right, let's put these 2 biological heavyweights head to head. If you need a quick cheat sheet for everything we just talked about, this is it. The contrasts are super stark. Mitosis is running exclusively in your somatic cells to keep the body functioning, while meiosis is strictly for your germ cells. Mitosis does 1 single division to make 2 identical cells. Meiosis pulls a double division to make 4 totally unique cells. Mitosis keeps the chromosome count perfectly flat at 2N, while meiosis slashes it down to N. Ultimately, mitosis is all about growth and repair. Meiosis, it's entirely about mixing up the gene pool and driving biological variation. As we wrap up this explainer, I really want to leave you pondering a wild thought. Your very uniqueness, your eye color, your weird quirks, your specific talents, it all relies on this microscopic genetic shuffle happening deep inside a single cell. Without meiosis, without that brilliant, messy genetic remix, we would literally all be identical clones, generation after generation. So tell me, what would a world filled entirely with identical human clones actually look like? How would humanity even survive a changing environment without that crucial genetic variation? Let that sink in for a bit, and until next time, keep exploring the microscopic wonders inside you.